our online class on glycolysis. This lesson is meant for undergraduate students in departments of medicine, natural science, biochemistry, nursing, and A-level students. Okay, so in the last two episodes, we looked at carbohydrates and we talked about glucose. By way of recap, we should remember that glucose is made up of 6 carbon atoms. So it's C6H12O6. Right, so that is the formula for glucose. If I represent it in the features projection, I'm going to do like that. Okay, so 2, 3, 4, 5, OH, this is D glucose. This is D glucose. Okay. In today's lesson, we're going to learn what glycolysis is. We're going to look at the steps of glycolysis. We're going to look at the reaction. So first of all, glycolysis is a metabolic process. Okay, it's a metabolic process where glucose is converted to pyruvate. That is the end product. Okay, where glucose is converted to pyruvate. Now, this process glycolysis takes place in the cytoplasm. Cytoplasm of all cells. That's where we have the enzymes of glycolysis. Okay, so glycolysis is divided into two important steps. The first one is the energy investment stage. Energy investment stage. Okay, and the second one is the energy production stage. Energy production stage. So, an overview of glycolysis. There is an investment of 2 molecules of ATP, and for the energy production stage, there is 4 molecules of ATP being produced. So if you pay back the ATP that was invested, so you have 2 ATP produced. Produced per molecule of glucose. 2 ATPs produced, okay, net per molecule of glucose. So it's not a very efficient method, but it's very important for life. We're going to see why that is. Aside from ATP production, there is a production of NADH, nicotinamide adenine dinucleotide hydrogen, which is used in the electron transport chain and oxidative phosphorylation to produce more ATP. We're going to look at that. It's a different process on its own. It is coupled with both glycolysis, the citric acid cycle, and oxidative phosphorylation. Okay, so let's go back to glycolysis. The first step in glycolysis is the conversion of glucose into glucose 6-phosphate, written as G6P, glucose 6-phosphate. This is the first energy investment stage, and we write it like this: ATP is invested, this is to yield ADP, which means that a phosphor group has been attached to the glucose to form glucose 6-phosphate. Now, because the phosphate group is attached in the 6th position, that's why we call it glucose 6-phosphate. Okay, this is substrate level phosphorylation. It's an example of substrate. This is a substrate level phosphorylation, and the enzyme that carries it out is called hexokinase. Okay, now glucose 6-phosphate is isomerized. Okay, is a reversible process by phosphoglucoisomerase, PGI, phosphoglucose isomerase, to form fructose 6-phosphate, that is F6P. Now, it's important that I mention at this stage that glucose 6-phosphate acts as an allosteric inhibitor of hexokinase. What that means is, if there is a lot of glucose 6-phosphate being produced, it will inhibit the action of hexokinase so that more glucose will not be funneled into this pathway. It will go into another pathway, which we're going to look at. Okay, we're going to look at gluconeogenesis, glycogenolysis, glycogenesis, and so on and so forth, which we're going to look at. Okay, so fructose 6-phosphate is an isomer of glucose 6-phosphate. Okay, now fructose 6-phosphate is now converted into a high energy compound called F16BP, which is fructose 1,6-bisphosphate. Now, it's important that we understand that it's not biphosphate, it's bis, high energy compound. Okay, and the enzyme that does that, this is the second part, that ATP will be used for ADP. Okay, so it's the PFK or PFK1, which is phosphofructokinase 1 or 6-phosphofructo 1 kinase, depending. okay, is essentially talking about the same thing. So this substance, okay, will now be broken into two by an enzyme that uses water to break them. All right, it's called an aldolase. So what I'm going to do, because of space, I'm going to wipe off this part. Then I'll start from F1,6-BP, fructose 1,6-bisphosphate, which will be cleaved by aldolase to form G3P and DHAP, which are isomers of each other. Okay, so aldolase breaks up fructose 1,6-bisphosphate to form glyceraldehyde 3-phosphate, which can isomerize to dihydroxyacetone phosphate, DHAP. And this enzyme here is triose phosphate isomerase. That's triose isomerase. That is the enzyme that catalyzes this isomerization reaction between glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Now, this is a 3-carbon compound. This is a 3-carbon compound. From a 6-carbon compound, we've now come down to 3-carbon compound, 2 3-carbon compounds. All right. So this one, glyceraldehyde 3-phosphate, will now, let me draw an arrow, will now yield through a reaction that is catalyzed by glyceraldehyde 3-phosphate dehydrogenase. So you have NAD plus to give you NADH plus H. Okay, nicotinamide adenine dinucleotide hydrogen. What this symbolizes is that hydrogen is like this. Okay, so it has been reduced, and from your knowledge of chemistry, from redox reaction, they are opposites. So if this is reduced, okay, by addition of hydrogen, it means that this will equally be oxidized. Okay, produces the oxidizing agent, and this is the reducing agent. That's okay. So this will be reduced, sorry, this will be oxidized to form free phosphoglycerate. Free phosphoglycerate. Remember, the enzyme here is glyceraldehyde 3-phosphate dehydrogenase, which is G3P dehydrogenase. This is how we write it. Glyceraldehyde 3-phosphate dehydrogenase, okay, which yields free phosphoglycerate. Now, free phosphoglycerate can to 2-phosphoglycerate can isomerize 2-phosphoglycerate. Okay, by the enzyme phosphoglycerate isomerase. Okay, so that is the enzyme here. So there's not enough space. Let me leave it. So this will be so this one 3-phosphoglycerate okay, I missed a step because this yields 1,3-bisphosphoglycerate. So I missed a step here. I missed a step there. So this, the product is 1,3-bisphosphoglycerate. So 1,3-bisphosphoglycerate will be converted to 3-phosphoglycerate. ADP, ATP. So this is the first production of ATP. Plus PI. Okay, so this is the first production of ATP. Free phosphoglycerate will now, the enzyme here is phosphoglycerate kinase. Okay, so that's the name of the enzyme. Phosphoglycerate kinase. The enzyme here is phosphoglycerate kinase. So this enzyme converts 1,3-bisphosphoglycerate to 3-phosphoglycerate with the production of ATP. 3-phosphoglycerate will now be converted to 2-phosphoglycerate. It's an isomerization reaction. Okay, and let me also say that here 2-ATP is produced. Okay, it's important that I make that clear. 2-ATP is produced. So here is reversible. Okay, so you have 2-phosphoglycerate. 2-phosphoglycerate. So, and is catalyzed by phosphoglycerate mutase. Okay? So that's the enzyme that catalyzes that reaction, phosphoglycerate mutase. So I'm going to write phosphoglycerate mutase. Okay, phosphoglycerate mutase. Okay, so then phosphoglycerate mutase is converted to phosphoenol pyruvate. Phosphoenol pyruvate, that process, you remember water is added here. So this place, water is removed. So I'm going to put this arrow here, PEP, what we call PEP. So we have the phosphoenol pyruvate. Okay, water is removed. Sorry, water is removed. H2O, and the enzyme is enolase. The enzyme is enolase. Okay, so PEP, which is phosphoenol pyruvate, will now be converted to pyruvate by the enzyme pyruvate kinase with the production of another 2 molecules of ATP. Now, someone will be like, where does this 2 come from? I'm going to show you here. So, phosphoenol pyruvate will now be converted to pyruvate. Okay, with the production of ATP, we have ADP plus PI, and the enzyme is pyruvate kinase. That is the enzyme. So, remember, it's still 2 molecules that is produced. Now, when you get to glyceride 3-phosphate, okay, which is cleaved into 2, remember, it's from 1 molecule of glucose. Now, glyceride 3-phosphate, okay, can isomerize to dihydroxyacetone phosphate, and it can change back. So, how we come about these 2 is that when you look at glyceride 3-phosphate and dihydroxyacetone phosphate, they are you can call them as 2 molecules, because there are 2 that have been cleaved into 2. So each direction, say for example, how I'm here on how we come about 2 molecules is from the cleaving of fructose 1,6-bisphosphate, which is cleaved into 2 G3P DHAP, so which can isomerize. So you can comfortably say that this one is now 2. This one is now 2 molecules because it can isomerize into this. Okay, good. So that is how we come about the 2. And each of those molecules going forward will now be 2. So when we get to the stage of say when we had NAD plus, like here, glyceride 3-phosphate to 1,3-BP, okay, in the reaction catalyzed by glyceride 3-phosphate dehydrogenase, where you have NAD plus 2 NADH, it's actually 2. Okay, it's 2. So from this stage, everything that is produced is in 2s. Okay, so that is why sorry. So when this kinase came in, okay, phosphoglycerate kinase, okay, when it came in here to form 3 phosphoglycerate, so it's actually 2 ADP to form 2 ATP. And you know this goes to 2 PG. So when this goes to PEP, it's actually 2 ADP to give 2 ATP. So that is how we come about the 2. So if you take a look, we have 2 here and we have 2 here. So in total, 2 ATP was invested and 4 ATP was harvested. That's why we say that the net yield of ATP in the process of glycolysis is 2 ATP. Okay? And then you also have 2 NADH. Okay? And then you have pyruvate. Okay? And then to also make it easier, it's still 2 pyruvate that is produced from 1 molecule of glucose. So these are the products. It's important that you remember that. So these are the products. In the next lesson, we're going to talk about the control point. Okay? We're going to talk about the control point. Then we're going to talk about the fate of NADH and pyruvate. Okay? Yeah, so in today's lesson, we've looked at the process of glycolysis. We looked at the products of glycolysis. And in the next lesson, we're going to look at the enzymatic control. Okay? And then we're going to look at 3 major points of control. Okay? So in next lesson, we're going to look at 3 major points of enzymatic control. Those ones are irreversible, where we have the hexo. We have the F16, that is PFK to make it easy for us, fructose 1,6 kinase. So PF phosphofructokinase 1, and then we have the pyruvate kinase. These are called rate limiting steps. These are the rate limiting steps, okay, or points of glycolysis. 3 major points of enzymatic control, rate limiting stages. Rate limiting. Okay, rate limiting. So that is what we've learned today, and that's what we're going to learn next lesson. All right. You can have a go and try to see if you can remember the different intermediates. Okay, I'm going to set a question. Try to see if you can remember it. What is the name of the enzyme that converts glucose to, what is the name of the enzyme? Okay, that's question 1. And question 2, what is the product when G6P is converted to okay, is the phosphoglucose isomerase. What does it produce? Okay, that's question 2. And then question 3. Question 3 is fructose 1,6-bisphosphate-BP is cleaved into two different metabolites. What are they? What are the 2 different metabolites? Which aldolase? Aldolase is the enzyme that cleaves fructose 1,6-bisphosphate into 2 metabolites. What are the names of those metabolites? Have a go at it and see if you can answer those questions. All right. See you again. Bye.